Hello, everybody. I'd like to welcome you to our monthly seminar talk hosted by the CDT Biodesign Engineering and organized by Dr. Naomi Nakayama. My name is Elizabeth Babarova, and I'm a PhD student on the CDT. I'm in my second year of my PhD and doing it in Dr. Patrick Jones' lab on synthetics and bioses. So this is our first event in the seminar series, which Naomi will be organizing throughout this year. And to kick off the CDT series, we have two speakers from the CDT, from the Imperial College team. That is Professors Geoff Baldwin and Professor Mark Isalan, who will tell you about the CDT and about their own group's research. First, we have a speak from Professor Geoff Baldwin, who is a professor of synthetic molecular biology at Imperial College London. And he is also the co-director of the Imperial College Center for Synthetic Biology, as well as the director of this CDT, the EPSRC Center for Doctoral Training in Biodesign Engineering. Research work in his lab focuses on synthetic biology approaches to facilitate the engineering of new biological systems for real-world applications, including the building of many foundational tools, such as basic DNA assembly. Geoff, take it away.
Thank you, Elizabeth, and thanks for the opportunity to talk to you today and to share some of the work that we're doing. If I can share my screen, is that up and running? Move that out of the way. Okay, so I want to talk to you today about the work we're doing around the accelerating the engineering of biological systems. So get my slides to advance. There we go. Right. So the technology platform that underpins synthetic biology is the engineering cycle, design, build, test and learn. So on the design side, as Elizabeth mentioned, we've done quite a lot of work around DNA assembly, creating the basic DNA assembly standard and automating that. And that gives us a framework for building things like biosynthetic pathways. The test aspect of this, really we're trying to include automation in the test aspect of what we're doing, and around the data that comes out of that, we'll also have quite a strong interest in metrology standards in order that we can normalize the readings that we're getting. And what I want to focus on today is some of the work that we've been doing around Learn, where we're trying to integrate AI approaches and active learning to actually really close the loop on the design, build, test, learn cycle. And one of the things that we've been working on and realizing as we've built out this technology platform is that really when you want to scale things, is that all of these things require underlying software and CAD frameworks in order that you can actually connect up these different aspects of the workflows. So for the build, go through this very quickly. So basic DNA assembly is a linker-based assembly. So we have parts in a standardized framework with a prefix and suffix with a type 2S sequence. So when we cleave that out of the storage plasmid, that means we have programmed different overhangs on each end of the DNA part, so we can ligate different linker adapters onto that. And then these oligo-linker adapters have a long single-stranded component, and that's what drives the assembly. So we call that the clip reaction. And so if we're doing, in this example, a 3-part assembly, we do 3 separate clips, purify those away from unincorporated oligos, and then we simply anneal them together before transformation. Ligase makes things worse at that step. So the key thing here is it's very modular and it's very standardized. Those linkers are key components of the assembly, but they're not kind of SCAR sequences per se. We have a variety of different types of linkers, neutral linkers that don't have any function, but they are also composable parts. So one of the key factors that we have are RBS linkers, where within the linker framework, we encode the RBS sequence. And we can also encode fusion protein linkers in order to build fusion proteins. And these aspects of the design framework proven very useful over the years in a number of different projects, and I'll be talking quite a lot about the RBS linkers. So here we can see, those linkers. So the upstream part here is the promoter, the downstream part is a fluorescent protein. So this is what we end up with, a promoter, an RBS driving GFP. And the linker overlap region here, and there's a little bit of space between, separates actually quite nicely the promoter from the RBS. Promoters in a design framework shouldn't actually be too close to the RBS, or you don't get efficient translation because you don't have a 5' untranslated region extending upstream of the RBS. So the linker here actually provides kind of a framework and a standardized framework that includes the RBS sequence. We now have libraries of these, so rather than using RBS designer software, we never bother with that. We simply sort of select from our library of known functional RBSs, and you can see here that we get quite a nice titration of translational regulation from using different standardized RBSs, and these are all standardized oligos, so we have them in the freezer. There's no synthesis required. We just go straight to build. So more recently, we've automated this process via our DNA bot software. So the DNA bot takes as input files which contain a description. They're just comma space variable files, CSV files, which have the list of what the parts are and their physical location on the plates. And similarly with the linkers, and also a constructs file which says what we want to make, and that's simply specified as the part name, linker name, part name, linker name, part name, linker name. And so that is fed into DNABot. What the software then does is it outputs 4 assembly scripts which can run on the Opentrons. We supply it with the part and linker plates, and it runs through the 4 separate steps of the process and delivers up to 88 constructs in a run, and also outputs some meta information. So that's working really nicely and has become actually the framework of how we do most of our DNA assembly in the lab these days. And since its original publication, we've actually now upgraded the DNABot software so it is all compatible with the latest version of the Opentrons hardware and API version 2. So this is an example of how we use that kind of automated framework. So this was what was in the DNABot paper. So here we have an assembly standard based on a 3-gene operon, so expressing 3 different fluorescent reporters, and we have 4 different strength promoters, and in front of each fluorescent reporter is a different RBS strength. So we have different UTR RBS linkers here, and we can vary the strength independently in each of those. And the other key advantage of this linker-based approach is we can change the gene order as well, because the order of the parts is defined by the linkers that we choose to ligate. It's unlike Golden Gate, where the order of assembly is defined by the entry vector, if you're familiar with that. So then this is kind of an output of that. So this is 88 variants of this design, where the basic reactions were spotted onto an SBS format agar plate and imaged for GFP, and then analysed by flow cytometry. And you can kind of see the degree of variation that we get as we're changing the design space here. And the key thing here really was, one of the key aspects of this was the accuracy of the process in delivering these constructs. So this leads us into a sort of plug and learn kind of framework, right? We now have modular DNA components. So here we have the promoter, the UTR RBS combinations, the gene of interest, and the backbone copy number. All of these can be varied within our framework. And this is the output just for a single gene GFP, varying, across 15 different RBSs and a couple of different copy numbers. And you can see it's sort of an order here, but it's not entirely consistent, right? The RBS isn't uniquely consistent as we change across these different factors. But one thing we did here was just using the JUMP software was to fit this to a statistical analysis. And the key thing here is that the parameters that we're fitting to are actually the DNA modules, right? So we're parameterizing based on the RBS, the origin of replication, and then the cross factors between the RBS UTR and the RBS ORI. And you can see if those are above the blue line in this, then they're significant factors in the fitting, and we get extremely good correlation. So this kind of demonstrates to us that we have fittable data based on parameters that are defined and are associated with the DNA modules. And this is important, right? Because it means that, you know, we have a consistent design framework. And so the way we think about this now is, you know, we developed basic assembly as a framework for building, so we could build much better and that we could automate the build process, and that's been very successful. What we realized and what became fully apparent once we started kind of working within these frameworks and fitting them is that that framework is not just a framework for design and build, but it's also a framework for learn. So that enables us to consider the unique components that are going into our design and to fit based on parameters associated with those DNA components. And so taking this forward, we're now sort of moving ahead with things that are things that aren't just fluorescent proteins. So we've been doing work with the violacing operon, and again, the basic framework lends itself to operon engineering, which is, you know, the way that microbes prefer to organize their genes, more efficient because the single RNA polymerase and single ribosome can create 5 individual proteins rather than requiring 5 separate promoters and 5 separate translation initiation events. So here, this framework, you know, we have 5 genes. We're varying the RBS of each of those genes individually and a range of promoters. And so far, you know, we've assembled 144 variants by DNA bot of this, and you can kind of see the output. We've now moved to post-transformation going into 12-well plates rather than spotting onto the SBS agar format, because we get more colonies and so we get more dependable transformation. And you can, you know, quite clearly see that the difference in output from these as we're changing the parameters. So the design space for this becomes pretty large pretty quickly, and sort of increasing the gene order or increasing the number of genes in your design framework, you know, gives you a large factor increase, because the gene order combinations is a factorial of the number of genes. Also, even with only 3 RBSs for each of the 5 genes, that gives a fairly large combination as well. So in total, just for this really quite constrained framework of DNA design, there's, you know, over 87,000 variants. So assembling 144 of these is pretty good, but it's only sampling a relatively small amount of this complete design space. Just to show you that we are sort of getting, you know, meaningful variations in output. You know, a small number of these we've taken through to kind of calculate the production of violacing in terms of mg per gram of dry cell weight of protein. So you can see that essentially the framework is working in delivering differences in output. But what we want to move towards is, you know, an AI augmented biodesign engineering framework. So we want to be able to use AI approaches to be able to learn a biological system and actually automate that biodesign process. So, you know, what we need here is that sort of design, build, test, learn type basis of the biological model, but we need to integrate our experimental and model data into a digital workflow and actually have active learning using AI to actually direct the experimental sequencer rather than the experimenter themselves doing that. So just a quick little bit of background on AI. This is obviously hugely prevalent in society now. It's kind of really coming of age. It's been around for decades. You know, AI in computer science started kind of back in the 1970s, but it's really only in the last decade that it's accelerated. And what led to this was deep learning and reinforcement learning, which kind of made some fairly staggering achievements. And the basis of this is to have an AI agent which can exert an action on your system, and it's then able to sense the environment, right? So it senses the state of the system, and on performing an action, it can compare now the state of the system with what it was previously. And if it's improved, that is, if it's closer to the objective that's been set, that you want the AI system to learn, then it gets a reward. And if it's worse, then it gets beaten with some kind of digital stick. And so the agent learns what gives a good reward through multiple iterations. And the key aspect of this is you can cycle around this very, very efficiently. By combining this with deep learning, where you have lots and lots of data, you can create very powerful learning environments. And this is what led to the development of AlphaGo, which in 2017 made a really big splash because it beat the world champion Go player. And this is a really challenging game because it's has much higher number of permutations in the game framework than chess does. So it's much more challenging AI problem to solve than chess even. The disadvantage of this is that it requires lots of data, and typically this deep learning aspect uses neural nets, which makes it harder to interpret. You know, we want intuitively to understand why something is better. And there was a key thing in the AlphaGo where I think it was at move 37, which was something that all of the experts, you know, master Go players couldn't understand what the AI system was doing. But then several moves later in the game, it became apparent that this was a genius move that kind of captured a segment of the board. So that's sort of advanced thinking, but it requires people to sort of look at the outcomes and try and interpret for themselves. The machine, you know, the code itself doesn't produce interpretable rules for people to follow. The advent of AI in biological sciences really, again, from DeepMind, not quite using deep reinforcement learning, but nonetheless, you know, using AI and deep tools, was its application to protein folding, which really made a splash in 2020 when they effectively solved, they would say, the protein folding problem to a large extent. And that really has dramatically changed. And I think what was interesting about that was it didn't require, you know, as a biochemist, I assumed that this problem would be solved through better force fields and, you know, better understanding of the basic physics, but it wasn't. It was deep learning based on really high quality structured data. And that's a key aspect of learning, is the quality of the data is really important in terms of the learning. And by this being based on, you know, the PDB, which represents decades of output of researchers and billions of dollars of investment. So machine learning approaches and applied to synthetic biology. So, you know, we've seen interest in, and I showed you a little bit of kind of DOE, statistical optimization. So that plug and learn fit that we had was really a statistical optimization. It doesn't give you any mechanistic information, but it's a very effective method. You can use this for design of experiment-based approaches, and it's a very effective method for navigating within well-defined parameters. And it can improve the efficiency of your experiment compared to random. The background knowledge really, though, is important, but it's kind of hidden. It's not explicit, but the way that the user defines the experiment is really important. Which parameters they choose to put into the DOE experiment will be dependent on how successful it is. Deep learning, which I've mentioned, really relies on the analysis of very big data sets, and so it's appropriate for genomics-based approaches. Key disadvantage is that typically it's black box, which means that it's not human interpretable. And it doesn't direct the experiment. It sort of is aimed at kind of learning the system, but it can't actually do active learning. So what we would like from an ideal AI approach is to have our background knowledge explicitly defined, have the code actually direct the experiment in an iterative way. So the code itself defines what the experiment is going to be. So in that sense, the user doesn't need to define the background knowledge. Importantly, it's better if we can have this working with sparse data sets. So, AlphaGo can play 40 million games of Go against itself. We can't yet, and probably never will be able to build 47 or 40 million different variants of a synthetic biology system and test them all to get that kind of depth of data. And ideally, we'd like the output to be human interpretable as well. So thinking about this, we kind of started looking at inductive logic programming, and this is based on inductive logic. So here we have some background information. A duck has wings, it can fly, it has a beak and webbed feet. And so we can generate a hypothesis that this bird is a duck. And so we can then go through inductive logic and ask a series of logic questions. Does it have wings? Can it fly, etc.? If the answer to any of those is no, then we have an uncogent hypothesis that that bird is not a duck. If we answer yes to all of these, we have a cogent hypothesis that it is a duck. But we have to remember that this is still a hypothesis. It might not be a duck, right? This is in fact a goose. And this is the basis of Karl Popper's falsifiability principle, which underpins the scientific process that we all use, whether we realize it or not, on a daily basis. This is how science advances, and is the basis of inductive logic. And from that flowed inductive reasoning, which is the principle of finding the simplest and most likely conclusions based on available observations. So the problem we have with this logic programming approach is that logic tests are based on binary output, and our experimental data, as you can see, is not binary. So what we've been doing in collaboration with Stephen Mulgerson and Wong-Jo Dai, a talented postdoc, is combining ILP with statistical regression. So the regression enables the numerical evaluation of responses, and the ILP gives an inductive framework for navigating our biological design landscape. So we can now ask, have a background knowledge in terms of things like promoter strength and RBS strength, and ask, hypotheses of does increasing RBS strength increase expression, and then by fitting the data, we can create an output to that to say yes or no. Advantage of this is the output is human interpretable. It can be used to drive experimental cycles, so the logic programming itself can generate the hypotheses to be tested, and it's also data efficient. So this is what we're trying to implement in our biological design automation. One of the issues is that we have a mixture of representation. So a genetic design is discrete. This is the input space for our 3-gene, 3-fluorescent operon. We can define the model based on a discrete hypothesis space by differential equations. So up to now we've been using the Weiss model to do that. But the output is time series, which is continuous. And so therefore it doesn't really match with the discrete hypothesis space. The deep statistical way of learning way of bridging that is to use a black box hypothesis, something like a neural net, which can bridge between those different types of language, discrete and continuous. But as we've said, that has impacts in terms of interpretability and the amount of training data required. Or the symbolic learning way is to discretize the outputs, right? So is the production under these different conditions good or bad, as we just said, so to convert it into a logic-based output. So these are the things that we are currently exploring in our approaches. So to do that, we basically build a model for the perceived values. We then translate that into logic facts, and from our knowledge base, we can then use symbolic reasoning and abduction to find the minimal kind of inconsistency and therefore to ultimately refine the model via supervised learning, and then we can iterate around the cycle, update the model and go again. So this becomes our perception part of the abductive learning, and this is the logic reasoning part. So the perception part requires statistical fitting and the reasoning requires logic programming. We're implementing this on synthetic biology targets. So design we do with DNAbot, we build that out, but then we're creating a digital twin from that model, and then we can compare our real world data to the model data. So the perception module carries out that data comparison, and then the logic reasoning can feed into an active learning component. So this is our AI agent, which can then direct the experiment. So this is what we're trying to build towards. We haven't done it yet, we haven't completed it yet, but this is where we're going with trying to use an AI-based platform to navigate biological design spaces. And as we move forward with this, currently we're working at the level of the genetic architecture. So these are the components that we're trying to optimize, but we've recognized that at some point, you can only get so far with modulating different expression of the components in the gene architecture. We want to kind of build out the machine learning curricula with biosynthetic pathways to lay enzyme kinetics on top of that. And then beyond that, to interact with actually the cell metabolic network, because if we're limited by the production of the precursors within the cell, then we need ways of perturbing this in order to advance. And so, using CRISPR-I and CRISPR-A to probe the cell metabolic network is the other place we're going with this project. So that's a very kind of swift survey of where we are in that project. Thanks to Gizem, Wangzhou and Liam, are kind of the key people working on that. As always, a large number of people kind of who've contributed to this work. So Marco and Matt were sort of really instrumental in my lab. Matt for sort of getting DNAbot up and running. Marco did a lot of work around the basic framework. And also, Stephen was a key collaborator in that project, or is a key collaborator in that project. So moving on, just to take a couple of minutes longer, just to introduce the CDT for you. So this started back in 2019, and synthetic biology had been conceived as the integration of engineering approaches to biology. And what we recognized when we were writing this proposal, that actually as the field was advancing, we really needed to bring data science into the training pipeline, right? Because the advent of high throughput technologies, the direction the field is going is that data science approaches are really fundamental in actually our ability to carry out the design, build, test, learn cycle. So we wanted to bring this solidly into the framework of our training program. So it's a partnership between three institutions, Imperial, Manchester and UCL. So we have an integrative training program. It covers experimentation, coding, data science, entrepreneurship, etc. And really the foundation for this was, captured best by this quote from Marcus at Synthase when we were writing the proposal. What the synthetic biology industry needs in terms of PhDs is lab competent coders and code competent biologists, both with a firm grasp of entrepreneurship and other business development skills. And that's kind of the framing concept that we had around the CDT, and it's still, I think, very much the mission statement that we live by on the CDT. So we have sort of, don't know if these numbers are still correct, but around 56 academic groups across the three institutions, covering really the full gamut of synthetic biology interests, from microbial engineering, mammalian engineering, cell-free protobiology, and also the control engineering aspects of the. So the structure of it is the students do our first year in systems and synthetic biology. They then go on to their PhD program. We have an annual conference. The annual training retreat has been somewhat interrupted by COVID, but we had our first one a few weeks ago, which was excellent. And we now have our monthly seminar program underway. So this is the training program as we're delivering it, and we have RRI kind of strongly incorporated across the whole thing, which is led by Phil Shapiro in Manchester and Jack Stilgo at UCL. So some of the key things that we're doing, one of the great things is, we've set up this student biofoundry with 6 Opentrons robots, and we are training our students in coding and automation. So they, at the end of in early December, students do a 2.5-week practical learning how to code Opentrons robots, and we're now seeing a number of students actually sort of take this facility on into their research projects and incorporating automation into what they do. As I said, a few weeks ago, we had our first annual retreat where we got some entrepreneurship training for the students, which covered all of the aspects that you need to think about in terms of turning an idea into a business opportunity. And one of the highlights of that week for me was Zach Sorrell's session on pitch training, which was truly inspirational and world class. And so I think the students had a real valuable kind of lesson from Zach there. That was amazing, and lots of other useful skills covered in the week. And at the end of it, the students were working in teams to develop a business idea and a pitch, which they sort of pitched Dragon's Den style at the end, and some really amazing ideas and pitches came out of that. It was really fantastic work done by the students there. So working with industry is a key part of the CDT basis. The idea is we're not training people to be professors, we're training people who are going to be the next industry leaders in the UK. And so reaching out and working with industry is a key part of that. One of the key things that we did with this was develop a pathway for working with micro and small companies where the company can pay a relatively small cost. Normally people would expect 50% to be funded by companies, but we reduced that to 5k a year, which for micro small startups is a more feasible level of funding, and we've seen a really good uptake on that actually, and that's been great. So our cohorts, we're just about to we've now recruited our 4th cohort, which will be starting in October, and we have one final cohort to go in 2023. So in total, there'll be 60 students who will be kind of co-funded by the EPSRC, and that's supported by both institutional and industry co-funding. They only give us enough money for 40, but we have to train 60. And so, with the industry funding so far, we've had 14 industry sponsored projects with a total of 389,000 actual cash contribution into that training, which is great. And 10 of those companies, fulfilled that small or micro definition that we came up with, and that's been really attractive to actually get startups into the CDT environment. So that's it from me. Apologies if I've run a bit over time. Lots to cover.
Wonderful. Thank you for that talk, Geoff. So we have a couple minutes for questions if anyone has any.
Can I ask a question? Oh, you have it. Then, so just quickly, the AI platforms you're building, Geoff, how generally applicable is it going to be for different kind of research projects, do you think, or even industry sort of?
I think the hope is that the the basis of the AI will be completely generic, as it were. You know, it won't be, it's not being coded specifically for, you know, violating biosynthetic pathways. I think, you know, what we're experiencing at the moment is sort of between the, you know, that statistical data fitting does require some kind of cellular model in order that you have to have something to fit to in order to then, you know, link that into the you know, the actual ILP symbolic learning module, and that I think is where the tension will be, because, you know, we started with the Vice model, we're adapting that and looking at different ways of thinking about the cellular model. But I think, you know, in terms of people who want to look at different organisms or whatever, you know, that's the piece that's probably going to be difficult in terms of porting it onto another kind of platform. But the core principles will all be, in principle, transferable.
Right. I think we have another question from Mark.
Hi, Geoff. Nice
Hi.
talk. I just wanted to ask you, because your vision of synthetic biology machine learning is really to do the machine learning in silico and apply it to the biological systems. But do you actually see a future for doing the machine learning inside cells?
So I think that depends what the problem is you're trying to solve. So for, you know, protein design, I think definitely, I mean, we're obviously seeing very powerful kind of in silico protein design now with AlphaFold and work at the Baker Group. But I still think there's sort of, you know, directed evolution inside cells where you're actually sort of observing the behavior of proteins in the environment where you want them to function, is still a very useful technology. Effectively, though, you know, if you're, what does it mean to do the design inside cells? You're probably then just looking at screening. Is that a posh way of saying screening?
Yeah, I think that's, you raised an interesting point, because I think directed evolution is a kind of learning, right? So it's just whether you can scale that and make it parallel and have learning units and multiple neurons, if you will, inside the system. I mean, we're working a little bit towards that. I'm just wondering if you're thinking along those lines too.
I suppose, I mean, you know, as you know, we're kind of doing some work in directed evolution, and I suppose what I'm kind of interested in is whether we can sort of have a hybrid in vivo kind of in silico where we're doing directed evolution and then generate kind of deep data, but then that sort of actually feeds into an in silico model as well, and whether you can then have that to sort of perhaps try and sort of jump forward. I mean, this has been a classical way that the sort of directed evolution has worked, right? You pick out 2 or 3 positive mutations and then you kind of like recombine to get your sort of generation 2, because that's actually more efficient than sort of asking the organism to go through all 2 or 3 mutant combinations because of the, you know, the problem of the exponential expansion of what you do. And so I suppose that, you know, the issue for me with the inside cell learning is, you know, it's hard to get away from that function of being reduced to how good is your screening methodology, which is never a particularly interesting question.
Right.
Right, we've got a couple more questions, but we want to have time also for Mark's talk. So I'm going to take one more question now, and if we could have the rest of the questions, you could stay back a little bit after the talks. I'm seeing a question from Oliver Hernandez Fernandez.
Yeah, that's a good pronunciation of my surname. Well done. I was just going to ask a bit of a general question for a lot of us who are just, you know, starting on their PhDs and stuff. I really like the way that you put data science into the whole spectrum of the engineering cycle. And I was wondering, it feels like there's so much that if you want to be in SynBio in general, they have to learn from all of these different fields, from just modeling, experimental work, automation, data science. Do you, on a spectrum, where do you think we should sit on whether we should just be complete specialists into one of those, whether we should be aware of all of them, or whether you think we should really go for being a jack of all trades, if that makes sense?
Yeah, no, I think that's a good question, actually. It's I do think you have to be, as a synthetic biologist, you have to be a bit of a magpie in terms of, you know, seeing some bright, shiny things and picking up on them and kind of cross-connecting them. So I do think there's always going to be a role for certainly being aware of the space. I don't think it's possible to be an expert in all of them. So I think, you know, collaboration and making connections is hugely important in modern science, and particularly in synthetic biology. But I do think you need to be really good at least one thing, right? So you know, absolutely become an expert in something, but try also to be not to the exclusion of everything else. You know, sometimes you get people who are expert in a field and they're kind of like, this is the way, it's the only way, and I don't hold to that. But so I think being really good at something, starting off with a real specialism is really good, but be open to everything and try and maintain a generalist attitude as well.
Jack of all trades, master of one. All right, thank you so much for answering those questions, Geoff. And thanks everybody for their patience. Mark, let's move on to your talk now. So our second speaker is Professor Mark Isalan, Professor of Synthetic Biology, also at Imperial College London, and he runs a research group in systems and synthetic biology in our life sciences. His main research interests are protein and gene network engineering, including gene circuits in bacterial and mammalian cells, answering basic questions on how to engineer patterns and structures, as well as more applied work toward human gene therapy. And what he's going to be talking about today is developing a genetic toolkit and gene switches to limit mycoplasma growth for synthetic vaccine chassis. Mark, take it away.
Thank you very much. I'll try and give you the fast version because I realise we're a little bit over time. So we've been working in my lab on moving beyond just E. coli and mammalian cell chassis. And we've been working on this wonderful beast that you can see in the middle, which you may have come across before. It's called Mycoplasma, and it's normally a contaminant in mammalian cell lines. But in this project, it's our main chassis for engineering. So mycoplasmas are really interesting, and they've actually been seminal model organisms for synthetic biology because they are actually the smallest free living cells. They have naturally genomes as small as about 500 or so genes, and artificial mycoplasma have even fewer genes. And so they've been a paradigm for the first synthetic life. There was this very famous paper from the Venter Institute where they chemically synthesized genomes of Mycoplasma genitalium and put them back into empty cell chassis in order to show that you could actually make a living organism purely at a genome level from re-chemically synthesizing its genome. Now, as well as being model organisms for systems and synthetic biology, these are actually important pathogens. In the laboratory, they attack our mammalian cell cultures, but in the wild, they are actually human and animal pathogens, and they have quite a lot of associated costs for the animal husbandry industry. And they're particularly difficult to treat with antibiotics because they don't have a cell wall and they have extremely slow metabolism, which makes them actually quite hard to kill. They're almost not alive in a way. But we have been part of an EU-funded consortium called MycoSynVac, which has purposefully tried to reuse the mycoplasma in order to do interesting biological engineering. The big vision of the consortium was to ultimately make a designed vaccine in order to target it to animal husbandry infections, for example, in pigs or cattle. And the way to do that was to strip the original mycoplasma of all virulence genes and to introduce into it just surface display vaccine epitopes that could be used ultimately to make a living vaccine. And our job within this €8 million consortium was actually to make biosafety circuits and kill switches in order to engineer into the mycoplasma to make safer vaccines. Now, that is a harder thing to do than it sounds, because mycoplasma are not E. coli, and there are actually incredibly few genetic tools that you can use in order to engineer in this organism. The natural mycoplasma only has 9 transcription factors compared to about 400 in E. coli. It's also more compatible with gram-positive machinery, and if you've ever engineered with gram-negative E. coli, the things that you're commonly familiar with just don't normally work inside mycoplasma. You also don't have plasmid systems, so we need to use transposons in order to deliver constructs into the cells. They integrate randomly, and then you screen out the constructs you want. So more important than that, there's very, very little stuff that you can use for the normal biological engineering. The parts that we're familiar with in E. coli, like inducible systems, arabinose operons, tetracycline inducible systems, LACI, etc., simply aren't available for mycoplasma. And so we set out to make a toolkit in order to do synthetic biology in mycoplasma, bearing in mind the fact that this is a very slow growing organism, whereas you can grow E. coli colonies easily overnight. It takes days, if not weeks, in order to grow your mycoplasma for each round of engineering in the lab. Its cell division time is around 24 hours compared to about 20 minutes for E. coli. So in order to build a toolkit, in order to start biological engineering in mycoplasma, the first thing you need are promoters in order to start playing around with the tools. So we were aiming to set up arabinose inducible systems, LACI inducible systems, lambda C1 controllable, repressible promoters, and also TETR inducible systems. And the starting point was just to screen for both synthetic and endogenous promoter systems that we could just introduce these operators into and see whether we could have basal activity from these systems. So what this graph shows you is that with an M-cherry reporter, we were able to get some promoter activity from these mixtures of different promoters, and they're all different strengths. So this purple one is quite a strong promoter, whereas this blue one down here is quite a weak promoter. But notice that this time of the experiment is 78 hours. So it's actually 3 days just to do a growth curve in which you're expressing a fluorescence and you can detect a system. But we were able to get these basal promoters. So then the next step is to then test these promoters in the presence of modified transcription factors, which are codon optimized and have various other changes in order to make them compatible with mycoplasma. So the example I'm going to work through now is an arabinose inducible system where you use the ARA-R arabinose receptor. It represses one of these promoters that I showed you on the previous page, and so that when ARA-R is expressed, it should repress cherry. When you add arabinose, it de-represses the ARA-R, so you should be able to induce red fluorescence. And that's exactly what you see on this bottom right-hand corner, that over a growth curve of the mycoplasma, you can see that when you add arabinose, and only when you add arabinose, you do see red fluorescence appearing. So in the blue curve at the bottom, you see the basal level of M-cherry fluorescence, which is very low, and only when you add the inducer do you get arabinose. So this works pretty well, and it's got a fairly low basal activity. And we did something similar for an IPTG inducible LACI system. So again, we had to pick different strength promoters that we could pretty much have zero basal activity, and yet when we introduce IPTG, you change the conformation of the LAC repressor, it stops binding its target operator on the promoter, and so you induce red fluorescence, which you can see in this curve on the bottom right here. So at the very bottom, I just make a few notes on how you have to optimize these kinds of systems to make them work from E. coli into Mycoplasma. So here we needed to play around with these synthetic promoters, and we also needed to put leader peptides onto the proteins. So a standard E. coli start codon isn't sufficiently effective to actually express the LACI protein. You actually have to put these small leader peptides in order to make them translate correctly, not just a ribosome binding site, but also these extra elements in order to make it work. So to cut a long story short, we carried on doing this for the 4 most commonly used inducible systems in E. coli, and we got fully functional arabinose, LACI IPTG, lambda C1, and tetracycline inducible TETR systems, all with fairly low basal levels shown in blue, and all with fairly good induction with their respective small molecule inducers shown in red in these curves here. So that's a toolkit at the promoter level. We also looked at the riboswitch level, and this is much more challenging in order to get this to work. But we did manage to get a riboswitch to work at least a little bit in Mycoplasma. And so a riboswitch, a very common one used in E. coli, is a theophylline one, and it binds to secondary structures of RNA in the leader of promoters of translation 5' UTRs, and either ribosome binding sites are hidden or exposed in the presence or absence of theophylline respectively. So when you add theophylline, you should induce expression of your promoter, and that's exactly what we see here. Unfortunately, the basal activity shown in blue on the bottom right is quite high, but at least when you add your theophylline, you can see the induction of this red curve up here. So we also got the ribosome switch working a little bit. So these are the tools. But then we wanted to do some gene circuit engineering in order to actually show that these tools can be used to make something useful inside the Mycoplasma. And our goal here was containment, biosafety kill switch, so that if you're building a vaccine and you release it into the environment, you could recall it, you could kill it in the absence of switches. So we built a series of kill switch circuits where you could either add components in order to kill the system, or in the absence of certain other components which you would provide in the laboratory, so when the Mycoplasma went into the environment, they would also die. So the example I'm going to show you here, which is kill switch 1, is a LACI inducible Cas9 with a guide RNA that cuts the Mycoplasma genome. There's no double strand break repair in Mycoplasma, so if you induce Cas9, you will get very efficient cell death. And so this is how our system is designed, with Mycoplasma components, using Mycoplasma promoters, parts of our new toolkit, which we put into this simple circuit. Now, in order to make this work, kill switches are very demanding because you're not allowed to have any leaky expression at all, or your system dies. So we had to find a particularly weak promoter for Cas9. We had to change the start codon from ATG to be a very weak CTG start codon, and we also had to do other things, such as put in suboptimal codons within some of the components in this network. But to cut a long story short, we were able to make this kill switch work, and you can see that at the bottom. And the only problem with this is that in the absence of guide RNA, your cells grow perfectly fine. This is a growth curve over time. When you induce your system with IPTG, your blue controlled growth curve switches into the red curve at the bottom middle that you see there with that red arrow. I keep losing connection. I think the battery on my mouse is a bit low, but bear with me, please. The only problem is that on the right-hand side, when you do this for more than one round, you start to see recovery of the Mycoplasma. and you start to get escape mutants which kill the Cas9 and become refractory and resistant to killing. So that is no good for a real world kill switch. Now, that was a polyclonal strain. When you switch to a monoclonal strain, which is purer, you get a slightly better result, but you still do get a certain degree of escape mutant frequency. And you can see that with these growth curves here, that your mycoplasma grow in the absence of any induction according to the blue growth curve. And when you introduce the kill switch, you end up with this red curve, which is fairly good, although towards the late stage you start to get escape mutants appearing, and when you've passaged the cells 15 times you get even more escape mutants appearing. So the stability is not yet good enough at that point, and your escape frequency is roughly something like 1 in 10 to the 4. We sequenced these in order to see what the escape mutants were, and they were things like truncated Cas9 and also mutations in the laci that prevent binding IPTG. So next generation sequencing really made sense to show why we were getting escape mutants in these initial kill switch models. So to get around that, we started to add redundancy to the system. We added a second kill switch, which we call KS2, and this one is controlled. In order to live, you have to add theophylline, which is not present inside cattle or in the environment. And in order to kill more actively, you also add anhydrotetracycline, a tetracycline analog. And in order to make the system work, it's repressed by tetracycline. 2 copies are required in order to make this work and keep the system switched off. And when it's induced, the tetracycline is de-repressed. It no longer represses a Cre recombinase, and then the Cre recombinase expresses and inverts a Cas9 in the system in order to make the system irreversibly switched on. So in order to make this work, we actually had to reduce the leakiness a lot. We needed 2 copies of TET-R, again a weak start codon on Cre and Cas9, lots of suboptimal codons on Cre, and the position on the polycystron also made a difference to our design. So the take-home message is, like in all synthetic biology, you really have to tweak the components in the system in order to make it work. And here we really had to tune them very, very weak in order to make really non-leaky kill switch systems. And I'm going to skip really quickly through the data of the kill switch, but the take-home message here is that you need to add theophylline, you need to remove theophylline and add anhydrotetracycline in order to induce Cre LOX recombination in order to activate Cas9, and it works very well. And in the final implementation, you have really undetectable Cas9 in the uninduced condition, which is very important to avoid escape mutants. So putting all this together, we made a double kill switch, mixing the kill switch 1, which I talked about previously, with this new kill switch 2 with the Cre recombinase system. And that way you have 3 levers to pull in order to get cell death. And it's much harder for the mycoplasma to get escape mutants. And indeed, this is the final data I'm going to show you. You end up with these very nice kill switch system, double kill switches, that when they're not induced, the mycoplasma grow happily in these blue growth curves. And when you do induce them, you don't get any growth of the mycoplasma. This drift is just the color of the pH in the medium. There's no cells here. And in fact, we have no detectable escape mutants in any of these systems. Not only that, but you can passage the mycoplasma for 15 passages. That's many weeks of passaging, and you still don't get any detectable escape systems. We also tested triple switches, but they were overkill and were in fact unclonable. So we think the sweet spot for these kill switches is double kill switches. So that's the summary of this genetic toolkit that I presented to you today. So we've got now inducible systems and riboswitches for Mycoplasma. We've shown that we can build these very stringent kill switches, and they're really quite strong, so strong that we think they could be applicable in the real world. I apologise for the speed with which I presented this, but we were running a little bit over time, and I know people had to leave at 5 o'clock. And if you want to read more about it, you can read about it in this paper which we just published this month in Nature Communications. This was part of an EC consortium called MycoSynvac, and nearly all of the work was done by Alicia Broto, who is a postdoc in my group. And I just want to leave you with the fact that the bigger picture of this project is that we were making vaccines. And if you want to actually hear a little bit more about this project, you can actually go to YouTube and Google this MycoSynvac video, which I can give you a very brief taster of, and you can hear more about this project. There are serious versions of the video and less serious versions. I'll give you a very brief 14-second version of the non-serious version, I think. Let's have a look at that. Okay, Google is being difficult as usual, giving me adverts, so I may have to skip that. But unfortunately, if you go to the site yourself, you'll see our nice MycoSynvac cow videos. So that's it. I'm very happy to take any questions now.
Thank you so much for that talk. Are there any questions for Mark? If there are any questions, please raise your hand.
So it's late in the day, and either I was very clear or completely incomprehensible.
I think it was clear.
Hands up. So as somebody who is setting up a new chassis for synthetic biology, I'm just wondering, do you have tips for you set up a new, system, basically a new chassis for SynBio too? Are there some sort of ways of thinking, sort of, any strategy that you can kind of pass on to the other people?
I think it's really hard. The best advice is talk to an expert who's already worked with that chassis in a biological context, if not synthetic biology. So for Mycoplasma, in this consortium, we had lots of Mycoplasma experts. So they weren't synthetic biology experts, but they could tell us about all the weird biology that was going on. And it's a really weird organism. As I say, some essential genes on average per cell have a copy number of RNA of less than 1 molecule per cell, and yet these are essential genes. It's incredibly stochastic and not intuitive to work with. So if you're working with a weird alga or a weird plant or bacterium, go and talk to people who've been studying that organism. And then the usual thing in synthetic biology, just be nice to your cells. If you're nasty to them, they will go into shock and do nasty things back to you. So be kind to them.
Yeah, knowing them, the humbleness actually is the best sort of going forward, isn't it? Thank you. Yeah, thanks. I like that.